Immunoglobulin antibodies consist of 4 polypeptide chains. Immunoglobulin antibodies, IgG antibodies, are large molecules having a molecular weight of approximately 150 kDa, composed of 2 different kinds of polypeptide chain. One of approximately 50 kDa is termed the heavy or H chain, and the other of 25 kDa is termed the light or L chain. Each IgG molecule consists of 2 heavy chains and 2 light chains. The 2 heavy chains are linked to each other by disulfide bonds, and each heavy chain is linked to a light chain by a disulfide bond. In any given immunoglobulin molecule, the 2 heavy chains and the 2 light chains are identical, giving an antibody molecule 2 identical antigen-binding sites and thus the ability to bind simultaneously to 2 identical structures. 2 types of light chain, termed lambda and kappa, are found in antibodies. A given immunoglobulin either has kappa chains or lambda chains, never one of each. No functional difference has been found between antibodies having lambda or kappa light chains, and either type of light chain may be found in antibodies of any of the 5 major classes. The ratio of the 2 types of light chain varies from species to species. In mice, the average kappa to lambda ratio is 20:1, whereas in humans it is 2:1, and in cattle it is 1:20. The reason for this variation is unknown. Distortions of this ratio can sometimes be used to detect the abnormal proliferation of a clone of B cells. These would all express the identical light chain, and thus an excess of lambda light chains in a person might indicate the presence of a B cell tumor producing lambda chains. By contrast, the class and thus the effector function of an antibody is defined by the structure of its heavy chain. There are 5 main heavy chain classes or isotypes, some of which have several subtypes, and these determine the functional activity of an antibody molecule. The 5 major classes of immunoglobulin are IgGM, IgGD, IgGG, IgGA, and IgGE. Their heavy chains are denoted by the corresponding lowercase Greek letter. IgG is by far the most abundant immunoglobulin and has several subclasses, IgG 1 through 4 in humans. Their distinctive functional properties are conferred by the carboxy terminal part of the heavy chain, where it is not associated with the light chain. And that's immunoglobulin antibodies consist of 4 polypeptide chains. Have a great day.